Hello, this is Miss Evelyn, again with the Veterinary Technology Program at CNM, and this is part 4 on microbiology, the fourth PowerPoint that goes along with VT 1096 clinical pathology, and again in the second week, part 4 on microbiology. The title on this one, or the topic, is to really present potential bioterrorism agents that would be of concern to veterinary medicine, especially since veterinary medicine is what we would call a sentinel. We are on the front lines of certain agents that might be used by suspicious people trying to do harm that would affect our animals first, and then it would come into the population of the humans. So that's what we would call it as far as bioterrorism agents or potential agents. Of course, these diseases can exist as their own diseases out there without any malicious side behind it. But we are concerned about these, and we watch out for these and employ appropriate PPE as we might be involved as veterinary technicians alongside our veterinarians dealing with the animal populations that may indeed be affected by these organisms. And this also will carry zoonotic concern organisms. So when we talk about agents of bioterrorism or potential bioterrorism, these would be bacterial agents that have been identified by the CDC, Center for Disease Control, as possible or probable bioterrorist agents. The reason for that is how they might be able to be applied into a group of animals or a group of people, and how that could usually progress with a rapid mortality, mortality kind of rate, some individuals may look at these agents for those kind of uses. Again, why is this important to us in the veterinary profession, and especially you as veterinary technicians in training? Because they can be there as potential agents. They can affect a variety of hosts, not just one species, but a variety. They tend to be easily transmitted. A high infectious rate with a low dose of the pathogen, and therefore highly pathogenic with high morbidity and mortality. So that's why these agents are really looked at. They include Bacillus anthracis, Francisella tularensis, Yersinia pestis, Brucella species, and Burkholderia species. Again, these are only potential agents, and these diseases do exist in our animal populations from time to time. So it doesn't mean if you see a Yersinia pestis case that it was because somebody was being malicious, but they again are potential agents of bioterrorism. And remember the reasons why. That they can affect a variety of hosts, be easily transmitted, highly infectious at a low dose, and also highly pathogenic with a high morbidity and mortality rates. We're going to look at these four agents, and again, you may have been introduced to these in another coursework at CNM in your animal diseases course. So this, in some case, might be a repetitive from there. But again, in the microbiology side, we're looking at how those organisms would look if they were grown out on media or being present to us in our laboratory specimens. So with Bacillus anthracis, and again, remember that when we have the first word is in capital, with our genus and our species is always small case, and that should be typed out in italics. Bacillus anthracis, or anthrax. A lot of times you will see this affecting the skin with skin lesions. It also can have an inhalation side, which is very deadly. It can be gastrointestinal, and we're talking about, again, some zoonotic concerns to humans, especially from infected meat. How does Bacillus anthracis look? It is a large gram-positive rod that forms spores, and it can live for long periods in that soil. It is endemic here in New Mexico, where we're located. There are parts of New Mexico where Bacillus anthracis is in the soil. It is because of the spores that are formed that it can really live long periods in that soil. And again, we certainly have a dry, low humidity climate that makes it very susceptible to the growth of anthrax. So the spores bring it into the soil, and then some causative environmental system maybe activates those spores, and we see it affect our animals. Again, reasons it could be utilized in a malicious manner is that various species, as you can see here, cattle, horses, mules, sheep, even goats, can really be infected and affected when they're grazing on grazing lands. Often we'll see para-acute with no symptoms, and the rancher wakes up one morning, goes out checking on his cattle, and he sees bloated carcasses out. And there's and the other part with anthrax is little or no rigor. And by that, there's no rigor mortis, there's no stiffening present. And so anytime that you would see dead animals with blood that doesn't clot coming out from from cavities, you need to jump right away and suspect anthrax. Now, most of the time you, as a veterinary technician, will be responding with your veterinarian, and it will be on their call that they are already thinking that we may have a potential really bad disease here, and PPE, appropriate PPE, should be utilized and addressed completely with different boots and foot coverings when you go into the soil area near a suspected anthrax case, and then those boots or shoe coverings should not come out of that area, not to your truck, not into a car, because that way you could be bringing the spores and anthrax to another area. So we have to really incorporate very good PPE while we're dealing with some of these organisms. You do need to know that anthrax can be called by wool sorter's disease. That can be a test question for you, wool sorter's disease. And the reason for that is it goes back to many years ago when humans would be handling infected wool from sheep or the hides, and those animals had anthrax, and the humans then got anthrax, and it was called wool sorter's disease because they were sorting the wool. In horses, it can be acute, 2 to 3 days to death. Blood again coming out of body cavities. So think about that. I probably should have blood out of body cavities highlighted on this slide. Just think, bacillus and blood, and that they would have really very slight to no rigor mortis being present. Dogs and cats tend to be resistant to the infection, but as you can see on the other column here, many of our grazing animals, cattle, horses, mules, sheep, goats, can actually be very deathly affected by it. Penicillin can work well for treatment. There is a vaccine for livestock in endemic areas. So here in the Southwest, we are probably vaccinating for anthrax, especially in very high prevalent areas. Again, anthrax will grow well on your BAP, your blood agar plate. Also a plate that's called chocolate, C-H-O-C is the abbreviation, but chocolate. It looks like a Hershey's candy bar. It is chocolate covered, and the media in that is the anthrax is very happy on that growth. So it grows on blood agar and on chocolate. Is a fast-growing colony, which has gray or white color. The edges of the colonies might be kind of curled and sticky. So remember how we identify the terminology that might be used to identify the morphology of how the colonies look, both to the naked eye on a agar plate, but also, of course, underneath the microscope. But these are all clues that if you suspect anthrax, Bacillus anthracis, how you're already looking at how those colonies grew on your agar plates. They call it a ground glass appearance. Ground glass. If you just can think of, if you took glass and you just ground it up and it looks kind of coarse and kind of clear, oh, that's a little bit what these colonies would look like. It is non-hemolytic. It is not motile if you do a motility test, and it is, though, catalase positive. So again, we have a large gram-positive rod that forms spores, and you might see those spores in the staining, non-hemolytic, non-motile, and the catalase is positive. Definitely, if you ever suspect anthrax is present, do not handle the carcass without proper, appropriate PPE on. The carcass should be burned and or buried. They usually will not even haul these away because they do not want the spores to potentially get in the environment and blow around elsewhere. And the herd should be quarantined, and you want to prevent other animals from scavenging on that carcass. And again, you may well have been introduced to Bacillus anthracis in your disease course. This section in microbiology is more concerned about how the organism would look on your in your microscope, how it grows on the agar, and just a couple of the safety notes here for handling a potential organism. The next one, tularemia. Again, this is also endemic in certain areas of New Mexico. So endemic, E-N-D-E-M-I-C, means that an organism is in the environment in this area, just like Lyme's disease. That organism is prevalent on the East Coast, and yes, it is getting in some other areas of the nation, but especially in Connecticut, New York, that upper East Coast area, Lyme's disease is endemic. So tularemia, Francisella tularensis, Francisella tularensis, tularemia is endemic in New Mexico. We are lucky to have that also here, or maybe unlucky, as you may want to look at it. The primary host is domestic sheep. It's often fatal, especially to lambs. So if there is tularemia present and there's lambing season, your rancher may call in that they are being affected and seeing many abortions happening or lambs dying right upon birth. Symptoms of this, of course, can be fever, diarrhea, weight loss in the animals, excessive urination, very rigid movements, and maybe they tend to isolate themselves from the flock. So those are some of the symptoms or signs that we would see. Tularemia can infect dogs, not usually being too serious, but cats, it may or may not be as serious, and pigs and pigs can be infected, horses not as common. But we do see it in our small animal medicine. And again, an alternate name for tularemia is called rabbit fever. So you need to know these alternate names because that might be what your veterinarian says, hey, we have a suspicious rabbit fever. And you need to understand that that means tularemia. So the wild animal hosts of rabbits is one of the primary hosts. And again, we call it rabbit fever because people can become infected if they are skinning rabbits. Beaver, muskrat, and mice can also carry this organism. It is spread by ticks, so tick control is very important. There is no vaccine. So again, it is spread by ticks around the rabbit burrows or rabbit holes. Rabbits might live a little bit with it, or if they die, and that's where our dogs and cats come into play. If they go out and they are around the rabbit burrows and they get bit by the tick that is carrying tularemia, and they can become a positive case. How does tularemia, Francisella tularensis, look? Under the microscope, it is a very small, faint-staining, gram-negative rod. They kind of call it, when you look under the microscope, that it looks like a rash. So if you can just picture reddish little rash, gram-negative rods, very tiny, very faint staining. It can produce a capsule, which again allows it to live for long periods of time in mud, water, decomposing materials, including the carcasses of those dead rabbits or other animals. Highly virulent, it can aerosolize. We can find it in wounds, lymph node aspirates, sputum. Again, it needs a cysteine-enriched agar to grow. So it needs cysteine-enriched agar, such as our chocolate plate again, something called TM and BCYE. So it needs that to grow. It is a slow-growing one. This is one that if you look at it at 18 and 24 and even 36 hours, you still may not see any growth. So if you're suspecting tularemia, you want to let it grow a longer period of time. The colonies may look gray-white, sort of opaque, smooth, shiny, as the colonies are there. Again, no hemolysis on a blood agar plate, no hemolysis. And they would be oxidase and urease negative. If you run your oxidase and urease tests, you will see that those are negative. You might have a weak catalase positive test, and it might be spread by that infected ticks, wood ticks, dog ticks, deer flies even. And again, there is no vaccine. So do remember, off of this slide, primary host is sheep, can really affect the pregnancies with abortions or death in young animals, quick death. The animals that it might infect, it's called rabbit fever, a very small, faint staining, gram-negative rod. Yersinia pestis. So we've already covered Yersinia pestis in our previous PowerPoint number 3. But a couple other points about it, and again, it's listed here because it could be a potential for bioterrorism. With Yersinia pestis, it can live up to about an hour in the aerosol or distributed in the air. Again, it's endemic here in New Mexico. So we're noticing that a lot of these organisms are here in New Mexico. They are in our wild rodent and rabbit populations, and unfortunately our domesticated animals, dogs and cats, and then also in times the grazing animals will get next to those infected areas. So it is endemic in New Mexico. Wild rodents and again rabbits are certainly affected. Rapidly lethal in cats, not so lethal as fast in dogs, but rapidly lethal in cats, especially if they get the respiratory form. And if the doctor is suspicious about it, streptomycin and tetracycline in combination can be effective against it. So you might see that utilized if you were in an animal hospital where they had a potential Yersinia pestis case. fleas. So with tularemia, we think ticks in the last slide. With Yersinia pestis, we think fleas are the primary vector. This one is a large gram-negative rod. And it is, of course, an Enterobacteriaceae. That was the family presented in PowerPoint number 3, Enterobacteriaceae. We call it a safety pin staining appearance. So it would be in that rod, it actually has sort of a rounded edges on either side. So if you follow my little cursor, I think is showing on your screen. And then on the ends, we call it safety pin staining. It would be darker area. Again, Yersinia pestis likes to grow on blood agar plates, your chocolate plates. Doesn't do as well on MAC as other Enterobacteriaceae, even though it is a gram negative, but it will grow on MAC. This one actually likes to grow at a little lower temperature. We usually incubate at 37 degrees Celsius. But this one actually likes a little lower temperature, so if you were suspicious of Yersinia pestis, you would probably want to set your incubator probably around 30 degrees Celsius or centigrade and see if it grew better there. This is why the organisms have been presented in week number 2 of microbiology, so that you have a little bit of an understanding what all these tests mean when they show up on these slides. So oxidase negative, urease negative, indole negative, all different tests that could be done, but it is a catalase positive. Okay, so it does like to bubble your hydrogen peroxide, your catalase test. And on the MAC, it does not, it's a non-lactose fermenter on MAC, on your McConkey plate, MAC. Brucella species. So there, again, in Brucella, the different species of animals tend to have their own Brucella species. So just Brucella species, brucellosis. Again, this could be used as a bioterrorism item. And again, of course, we see this as just an infection in some animals. Again, you need to know the alternate names. Bang's disease or Malta fever, M-A-L-T-A fever. Bang's disease or Malta fever. You do not have to memorize the different brucellosis species, but they are listed here. Brucellosis bilitensis in goats, obtus in cows, Brucellosis suis, that should make you think of pigs, suis, and canis in dogs. Mostly species specific, but they can cross over, but mostly species specific. And a lot of what we will see is abortions, stillbirth, breeding issues, epididymitis in sheep leading to sterility in rams, abortions again in pigs. But we see a lot of abortion and breeding issues with brucellosis. In fact, in a breeding kennel of dogs, most of the time you're going to have breeders test brucellosis before they would put a bitch and a stud next to each other to make sure that the animals are clean and not infected, because it can cross upon a breeding, and we do not want to have that affect a really prized animal. So cattle abortions, even in unvaccinated herds, organisms are shed in the milk and uterine discharge. One of the biggest things to remember, if you are suspicious of a brucellosis case, whether it is small animal or large animal, make sure you have appropriate PPE on, personal protective equipment, on before you would start handling a fetus or discharge from the vulva or discharge from the penile area, you are definitely going to want to have PPE on because, of course, this is a zoonotic concern also. Pigs, we can see septicemia, early abortions. They may not, the owners may not even realize that abortion is happening. Occasionally, we see a brucellosis case that shows a sign of a fistula on the withers of our horses. And they often, horses can get it from infected cattle. So what does brucellosis basically look like? A small, faint staining, gram-negative rod. And yes, unless you actually saw these, how could you know? It's really larger than Franciella. But that's how we kind of note it, okay? So a small, faint staining, gram-negative rod. It likes slow growing on our blood agar plates. It will grow on chocolate plates and on MAC. Small, glistening colonies, non-hemolytic, catalase positive, oxidase and urease positive also. So with brucellosis, remember it's called Bang's disease and Malta fever. Most of these are species specific, but they can cross over. A lot of abortion and other breeding issues can be signs that we will see. It can be shed in the milk, therefore could be passed to humans also. And it is a small, faint staining, gram-negative rod, non-hemolytic, catalase positive. Our last one in this list of potential bioterrorism agents is Burkholderia malii and pseudomalii. So Burkholderia malii and pseudomalii. Melioidosis it causes. So Burkholderia malii causes what's called glanders in horses. So remember, glanders in horses, contagious, usually fatal, upper respiratory tract. So it affects the glands up in the upper respiratory tract of your horse. Burkholderia pseudomalii occurs in many other animals and primates, can infect in many different ways, and it can look like many different things. I know that's very specific, isn't it? Just come back and remember, Burkholderia malii, glanders in horses, affects the glands, respiratory infection. And again, lung abscesses, pneumonia. It can cause mastitis in goats, and the organism can be isolated from their milk. You might see lameness, you might see encephalitis. Again, in horses, because they're under distress, they might break with a colic disease, they might break with diarrhea, and certainly respiratory and especially upper respiratory concerns, nervous system disorder. So Burkholderia malii is a gram-negative rod. It's a larger rod like Pseudomonas. Again, without seeing some of these under a microscope, you say, how do I know? But this is how we kind of, again, specify what they look like. It grows well on blood agar, chocolate, and MacConkies. It's a gram-negative, therefore likes to grow on our MacConkies, the MAC. Pseudomalii grows faster than the malii. Pseudomalii is oxidase positive. Malii can be variable. And again, pseudomalii is motile, malii is non-motile. Okay, so we're really concerned about this Burkholderia malii, and it's catalase positive. Remember, it's called glanders in horses. A lot of upper respiratory lung pneumonia situations, gram-negative rod, and likes to grow on our blood agar, it's catalase positive. So the next section we're going to go through, and you will see that there's several repeated organisms that you've already been introduced to. But these are what we call the veterinary technician zoonoses. Zoonoses. So these definitely can transmit from animals to humans. Pasteurellosis is our first one that we're going to mention, with Pasteurella multicidae, Canis hemolytica. So different Pasteurella species. This is the most common bite-associated infection, bacterial infection. You should remember that for test day. So pasteurellosis is the most common bite-associated infection. It's a normal flora in our dogs and cats, and it usually doesn't make them sick. And it's found where? In the oral cavity of healthy cats. Therefore, if you have a wound on yourself and your animal comes over and licks you, you might end up with a pasteurellosis infection, and they can be quite devastating to the tissue area and so forth. And again, in a bite wound, a lot of pasteurellosis, Pasteurella infections in humans. So Pasteurellosis canis, bite wounds in dogs. Hemolytica affects sheep and other ruminants. Again, you need to remember some of these little names. So pasteurellosis by hemolytica can cause what's called shipping fever in cattle. Now, other organisms may also cause this, but we do see this with pasteurellosis organisms. It is a small gram-negative rod, non-motile. Hopefully in your hands-on class time you will be able to see what we mean really by a bacteria being motile. It is really a neat experience when you can see that, the motility factor. So if you run that test, your pasteurellosis is non-motile, oxidase and catalase positive, and we call it a facultative anaerobe. So think about if you end up with a wound and a dog has bit you or caused that wound, and it closes over on the surface. Isn't that an anaerobic then area underneath your skin? Bacteria just really loves that. Pasteurellosis. Haemophilus influenza is a respiratory infection we see in pigs. Haemophilus hemolytica, it again causes a shipping fever in cattle. So a lot of your animals will get on the trucks for transport, are shipped out, and it's a very stressful time. They're thrown together, many animals in a cargo truck or shipping trucks, and they end up at a processing plant or grow out feedlot, and they are just under stress. So if there's any subclinical infection on, they may break with it. That's why we call it shipping fever. Okay, so different organisms might cause that. So Haemophilus and Pasteurella are in the same family. We have this is a small gram-negative rod. It's again non-motile, goes on chocolate, but not on blood auger or McConkey. So even though it's gram-negative, it does not like to grow on our McConkey. That's Haemophilus influenza and Haemophilus hemolytica, small gram-negative rod. A next organism on the lower part of this slide, Bordetella bronchoseptica. Bordetella bronchoseptica, if you say it with me. One more time, Bordetella bronchoseptica. We do vaccinate for this. It causes kennel cough in dogs, and it can cause a pneumonia in cats. It can be acquired by humans who work in very close proximity to the animals. So probably not if you just have one or two dogs around, but in a kennel cough situation, you might be able to get it. Sometimes we actually have in kennels, boarding kennels, they may have ultraviolet lights that help maybe with controlling some of this bacterial growth. It is motile. It is a gram-negative, what we call coccobacillus. It's sort of round, it's sort of oval, it's a kind of coccobacillus. It's halfway in between. Bordetella bronchoseptica, and again, we can vaccinate for that. Not all dogs are, but a lot of boarding kennels will require that. Salmonellosis, we've already covered salmonella in our previous PowerPoint number 3, but this can be caused by a variety of species of salmonella. It affects many different animals, and it may be septicemic. Now remember, we're talking about zoonotic concerns here, so diseases that can be passed from animals to humans. You might have an acute or a very chronic enteritis, so inflammation, itis, I-T-I-S, of the E-N-T-E-R, entero area, so inflammation of your small intestines. Again, this is a breakdown of medical terminology. The usual infection route is fecal-oral. Organisms can multiply in your intestinal tract. It is one of our Enterobacteriaceae by family grouping. It's a gram-negative rod that is motile. So just remember, and also reptiles is one of the main areas that salmonella, reptiles and poultry, that we see salmonella being a problem with zoonotic concerns to humans. Gram-negative rod, motile. Listeriosis or circling disease, again, another name that you need to know, might show up on a test, that circling disease is listeriosis. Most often with encephalitis or meningioencephalitis in our ruminants. It's caused by Listeria monocytogenes. Listeria monocytogenes, maybe in silage or the hay that the animals eat. So remember, this is bacterial and enters from cuts and abrasions in the mouth and then travels to the brain. Doesn't that sound like an ugly transmission? Affected animals animals might be seen with fever and depression, incoordination. They might segregate themselves, again, isolate. They might lean on a fence or a wall because this is affecting their brain and their meninges. Okay, they might start circling in one disease, and that is why it is called circling disease, because they might start circling in one direction as their brain and meninges are affected. It can also cause septicemia in monogastric animals and young calves. It might cause abortion and stillbirth, mastitis in ruminants, and the organism can be shed in milk, which is the zoonotic concern to humans. That if a dairy cow has this and we get that milk into our human pipeline, we can end up with listeriosis outbreaks, very serious diseases then once it gets into a human. And the organism can also cross the placenta and infect the fetus. Again, this is a small motile. It has a tumbling motility. Tumbling motility, remember that. Gram-positive rod, but a small motile tumbling motility gram-positive rod. Grows well under refrigeration. So what does that tell you about this problem that it can be shed in the milk? We go and refrigerate our milk, and therefore that organism is just loving that. So you would want to incubate a potential listeriosis at definitely a lower temperature than 37 degrees centigrade. Okay, into clostridial diseases. Okay, so Clostridium, you were probably again introduced to this in your animal disease course last term, but Clostridium, when you look at it in the microbiology side, it is a large, very large, gram-positive, spore-forming anaerobic rod. So large gram-positive spore-forming anaerobic rod. That's why it causes our gas gangrene, is because it would be present. Some of these Clostridial agents can be present on rusty nails, barbed wire fences, claws of animals, of cats or something like that. you get a puncture wound that closes over very rapidly on the outside layer of your skin or on the animal's skin. And then this Clostridium that is anaerobic, loves to grow in a non-oxygen area, starts growing and prolificating, and that's where you get the gas gangrene kind of situations. It's that gas underneath the tissue that is really causing the problems, and there's producing of toxins. Sometimes those toxins can be ingested, and the toxins, instead of the organism, are responsible for the disease state. So a couple different, so Clostridium in general, large gram-positive spore forming. You might see spores in these when you are gram staining them. So look at your book again in how those spores be present. So Clostridium perfringens causes that gas gangrene. It can cause hemorrhagic enteritis in dogs, dysentery in lambs, and enterotoxemia in piglets, foals, and goats. So Clostridium perfringens can cause a lot of enteritis, hemorrhagic enteritis, dysentery, another nice word for diarrhea. Clostridium botulinum causing botulism. This is actually by ingestion of a toxin from a decomposing animal, carcass, or plant material, and it can cause what's called flaccid paralysis. Flaccid paralysis. The animals just can't walk right. Pigs and dogs tend to be relatively resistant to botulism. Cats don't seem to get it at all. Clinical signs can have difficulty chewing or swallowing, paralysis that can affect the heart and lungs and causes death. And again, cattle normally ingest these toxins in silage or contaminated grain. So contaminated grain to some of our grazing animals could be a problem. And signs can include drooling and a tongue hanging out, difficulty eating, and weakness. Okay, so Clostridium botulinum, botulism. In horses, Clostridium botulinum can do similar signs as in cattle. Horses develop forage poisoning from ingesting the toxins. And then there's toxic co-infectious diseases can come into play, wound infections. That's where we see also a lot of Clostridium problems with wound infections. And with birds, they can have paralysis of the legs, wings, neck, and even the eyelids. So a different Clostridium that we probably are a little bit more familiar with from our human side is Clostridium tetani, or tetanus. So this is bacteria that is introduced into deep wounds, produces a toxin, which causes rigid paralysis and is definitely zoonotic. And this is why within our veterinary technology program, tetanus is the one vaccine that we do ask that you have current within the last 10 years, because we can just come into Clostridium tetani quite readily off and on in the environment that we're caring for our animals. We are nearing conclusion on this PowerPoint. Erysipelas is caused by, that's why we call it erysipelas. Erysipelotheryx ruseopathy. I probably butchered that word. But erysipelas, slender gram-positive rod, can be easily over-decolorized. So if you are suspicious of this, watch your decolorization stage. You may only want to touch it once or twice in your gram stain process. Slender gram-positive rod. It is catalase negative, non-motile. This will live in the soil and the water and decaying organic matter and carcasses, multiplies in tonsils and lymph nodes, and gains access into the bloodstream. And the worst thing with erysipelas is septicemia in pigs, arthritis in lambs, laminitis in sheep. They might go through a sheep dip, and if that organism is present in the water or around the edges of the water that the sheep go through, they might get infected and they'll get laminitis in their hoofs. But septicemia in pigs, erysipelas. Leptospirosis, we basically have two more organisms. Leptospirosis is one of those. Leptospira interrogans. So leptospirosis, spirosis should make you think of the word spiral. Basically, anytime you see a spirochete in your microbiology slides on organisms, spirals tend to be bad. You really don't want a disease present with spiros, and leptospirosis is one of those. There's over 200 strains, or as they call it, serovars, probably more than that at this point. Some are host-specific, others are not. And this is one of the most common zoonotic diseases in the world, especially in other parts of the world where human waste and urine and animal urine is not as well kept out of water sources. So Leptospira interrogans. They call it a helical rod, but it's really a spiral. So a little bit of a spiral. You should find a picture about that. This would be an interesting one to see what that looks like. It's excreted in the urine. And again, leptospirosis is sometimes tested for, or wanting to make sure that that the male and the female dogs do not carry that disease and be passed during breeding time. Excreted in the urine, this is also where we see our dog habits of the male dogs wanting to go and sniff at a fire hydrant, urinate, and the next dog comes along and sniffs and inhales the leptospira and gets the disease. That's how they can pass it. Or they lick each other's private parts, as disgusting as that might be. So anytime there's contact to urine or in the water where an animal urinated, they might be able to get this, you might get the disease passed. Drinking contaminated water. It's distributed through the bloodstream to all of the tissues. It causes kidney and liver damage. It might cause abortion and stillbirth. That is why we're concerned about breeding colonies, especially in our canine world. May pass it from to man from pigs. So Swineherds disease, it's called Swineherds disease. In horses, they call it periodic ophthalmia or moon blindness. Periodic ophthalmia or moon blindness. Cats don't usually show any problems. Continuing with this, part of that is because the inflammation in the middle layer of the eye is very painful, sensitive to light, waters excessively, eyelid muscle spasms, and that will help keep that eye closed. That's with our horses. Dogs, we might see fever, vomiting, pretty high fever, vomiting, diarrhea, abdominal pain, bloody urine to all the way up to kidney failure. This is a very severe illness to a dog. We used to almost always vaccinate for leptospirosis. Now it is one of those diseases that we may vaccinate at times, especially if it is a breeding colony, an animal that is being shown a lot. If you take your dog all the time to a dog park where getting next to other potentially infected urine might be a problem. But it also, as a bacterian vaccination, is the one that caused a lot of stinging to our animals and sometimes even superficial irritation at the site of that vaccination. So some veterinarians have moved away from including leptospirosis as a routine vaccination for dogs, but it is there still in our dog world And maybe if you were in certain areas in the nation, it might be one of those routine vaccinations. With cattle, acute or chronic conditions, mostly in young calves, again, chronic abortion, stillbirth, so again, related to breeding issues. There is a vaccine that's available. They call it multivalent for cattle, pigs, and dogs, but not for horses, and no vaccination for humans if we get it. So think about abortions. Our last slide and our last disease organism on our journey through organisms. Ehrlichiosis, or Ehrlichia, Ehrlichia, Ehrlichia species. This is a small gram-negative, they call it obligate intracellular coccobacilli. This infects, unlike a lot of our other organisms, this one actually infects the white blood cells. It is tick-borne disease by the tick, and it may infect your monocytes and cause a monocytic ehrlichiosis, or neutrophils with a granulocytic ehrlichiosis. So this is actually a bacteria that gets into the white blood cells. It is tick-borne. It is a small gram-negative. Dogs may actually have various signs, might develop subclinical or chronic. They may show absolutely no signs, but the serum proteins, when you get into hematology in your study of in next term in our coursework, you may actually see some changes in your serum protein might be increased. There are some antibiotics to be able to treat this. Horses, the disease progresses to severe over several days. Again, symptoms might be incoordination, reluctance to move, and edema in the hind legs. Cattle and sheep, most recover spontaneously, but stress, again, if they were shipped, you might see a relapse on them from a condition of ehrlichiosis. Remember, it's small, gram-negative, obligate intracellular coccobacilli, again, a little bit round, a little bit like a rod, and it affects the white blood cells. Tick-borne, remember those parts. With this conclusion of this slide, we have ended our journey of the organisms for VT 1096, the microbiology section. Go back, review the material, match it up to your textbook, The Laboratory Procedures for Veterinary Technicians by Margie Sirios, and enjoy studying these organisms. Thank you very much.